DNA polymerase can't start DNA chains de novo. So it can't synthesize DNA from nothing. Even if you give it a template, DNA polymerase is incapable of making a DNA polymer. So if you give it a single-stranded DNA, it's unable to catalyze synthesis. What it requires is a primer, and that primer can either be DNA or it can be made of RNA. So this, of course, raises a question. Where's this primer coming from? Who makes the primer? How is that regulated? And, just as an aside out of curiosity, can you think of an instance where if I supplied a single-stranded DNA to DNA polymerase, it could actually synthesize DNA without a primer, without a second molecule? So one example may be if you had a DNA that was able to form a hairpin. So if we had something that came around like this, and we had some base pairing going on here. So if it formed a loop, okay, for example, what's really important here is the 3' hydroxyl. And we'll get into some details of that in a moment. But of course, if this DNA chain, I guess, is going to go 5' to 3' in that crazy anti-parallel fashion, that this 3' hydroxyl has some propensity to be able to form the secondary structure that we see here. If it's basically self-complementary, that you could actually provide some sort of equivalent of a primary, even though it's the same molecule. And this could facilitate catalysis in some way, just sort of an aside. So another thing that they found was this enzyme was also unidirectional. That is, it only synthesizes DNA in the 5' to 3' direction. Okay, and as you all know, we have two strands of DNA, one running 5' to 3' and one running 3' to 5'. And so in the context of double-stranded DNA, the enzyme would be presented with one template going in the right direction and one template that's going in the wrong direction. So question becomes, how do we copy the other strand? Okay, so how to copy the other strand? Okay, how does that happen? They also found that the enzyme was not particularly processive, nor did it catalyze its polymerization reaction rapidly. So what I mean by processivity is the number of nucleotides that it synthesizes per template binding event. Okay, so number of nucleotides synthesized per binding event. Okay, so you might imagine if you say you're replicating an E. coli chromosome from one replication origin, which is the case in E. coli, you might want that to have that enzyme that once it binds and starts polymerizing, to be capable of synthesizing thousands of base pairs before it dissociates from the template. If you have something that, ping-ponging off and on, or make a couple of dissociate rounds and synthesize a couple, dissociate, synthesize a couple, and so on, that's not going to be anywhere near as efficient in the synthesis. And of course, it's not going to be rapid. So the observation that Pol 1 was not highly processive was sort of curious, especially in the context of what they were expecting it to do in vivo. And then the last curious thing that they found was no matter how much they purified and fractionated their DNA polymerase, it always had an exonuclease activity associated with it. And so an exonuclease is an enzyme that degrades DNA from the end. If it were degrading in the middle, it would be called an endo. So it degrades DNA from the end. And the interesting thing again with this was that it also had polarity. So it was a 3' to 5' exonuclease. So again, so this was very curious because the polymerase is synthesizing 5' to 3', and then you have a very tightly associated exonuclease that is degrading 3' to 5'. So which, you know, seems a bit counterintuitive. So they were, of course, very curious about what this exonuclease activity could possibly be for. So let's now have a look at what's going on with the reaction. So this is a bit of a schematic, and what we have here is basically a template, a DNA strand, and I guess over here we have this short primer. Okay, and as I mentioned, the primer is providing a 3' hydroxyl here. So it is this part here, so 3' hydroxyl. And that 3' hydroxyl is critically important because this is what catalyzes or conducts a nucleophilic attack on the alpha phosphate of this incoming nucleotide. So the nucleophilic attack is here, and this is our alpha phosphate of that incoming deoxynucleotide. And coincident with that, we get the formation of the bond and the leaving group, which is pyrophosphate. So we can think of PPI being released, which is this here. So this is the beta phosphate and this is the gamma. And we can call this pyrophosphate. Okay. So a couple things worth pointing out here. Once you finish the first cycle of the reaction, so one polymerization event, the product of it has a 3' hydroxyl. Again here, that's poised for the next reaction. So we have another free 3' hydroxyl for next reaction. So that's kind of nice. Your product is actually a substrate for another reaction. So a very simple reaction. Question then becomes, what drives this reaction forward? So we could write it in a sort of simplified way. We have a DNA polymer of some length, so we can call it N. So if we write this down here, DNMP to the N, okay? And then we give it a deoxynucleotide with a DNTP. We now get a polymer that's now N+1, so DNMP N+1, okay, and pyrophosphate, PPI. Okay, and like all chemical reactions, it's reversible. So I'm sure you can appreciate that usually this reaction runs forward towards polymerization. So the question is, how does it go towards polymerization? Is there anything in particular that you can think about for PPI? Well, the reaction is favored because of the cleavage of the two high energy bonds, right? So the first one, so is where, you know, the pyrophosphate that leaves. So we get cleavage here. So this at this point, okay. And then the second one is actually forming the cleavage of PPI, so pyrophosphate. So simple hydrolysis reaction, we can get two inorganic phosphates. Okay, and this, the enzyme that catalyzes this reaction in cells is done by an enzyme called pyrophosphatase. And that's a hydrolase enzyme. So the cleavage of both of these phosphoanhydrate bonds drives the reaction forward because they're both high energy bonds. So one from the initial nucleophilic attack of the 3' hydroxyl on the incoming DNTP, so this one here, and then the second one from the subsequent cleavage of pyrophosphate. So if this is cleaved again, number 2 to 2-pi. So what's another consequence of cleaving pyrophosphate? What would you actually need to push the reaction backwards? Because you could manipulate this reaction to make it run in reverse, right? So what would you have to give it? Probably like high levels of pyrophosphate, right? Because the reverse reaction, for the reverse reaction, one of the substrates is pyrophosphate. So this one here. But if the cellular concentration of pyrophosphate is always essentially 0, which it is, there's really no way you can push the reaction in the reverse direction. Okay, so if you wanted to reverse it, reverse reaction, need high levels of PPI, but we have low concentrations of PPI in cells. So what essentially this does, it decreases the possibility of the reverse reaction. All right, so basically there's no way to push the reaction in there in the reverse direction. So the existing low concentration of pyrophosphate is actually a factor in promoting the forward reaction, or at least preventing or prohibiting the reverse reaction. So what other reactions are driven by pyrophosphate hydrolysis that you may have that we may have encountered recently? What about in the nucleotide biosynthesis lecture? Remember how we talked about the condensations that we saw with PRPP when we're adding bases to the ribose? So it's the same type of thing. That releases a pyrophosphate, and that's driven forward in part by hydrolysis of pyrophosphate. We'll meet other examples later on, and again, for just to give you an idea, tRNA charging, but I won't give anything away at this point. So it's really a theme in biology that you can push reactions by pyrophosphate hydrolysis forward for reactions that actually release pyrophosphate. In keeping with the polymerization reaction, we did mention in the introduction that it's really super important that this reaction take place and be of high fidelity. So the question becomes, how is high fidelity achieved? Well, first of all, what does fidelity mean? Fidelity is usually expressed as an error rate, and the error rate is the fraction of the time that the enzyme inserts an incorrect base. Okay, so let's just define this. Error rate. And it's the fraction of time a wrong base is incorporated. So if you had a template that was a poly DA, you would ask how frequently does DNA polymerase insert something other than poly DT? And it's known that the in vivo error rate is typically about 1 error per 10 billion bases synthesized. So in vivo, we have about 1 error per 10 billion bases synthesized. So that becomes 1 over 10 to the 10, or 10 to the minus 10 is typically how it's written. Okay, so that's what we're aiming for. And I guess to describe this is, how do we get from random, which if random insertion occurred, we'd probably make an error 3/4 of the time, right? How do we get from 3/4 to 10 to the minus 10? So there's a number of contributions, and we're going to outline them on this slide. First, which is kind of obvious from the Watson-Crick model, has to do with the complementarity between the template and the incoming base. And this, of course, is dictated by the hydrogen bonding between the base on the template and the base that's entering. And that accounts for about 1 in 100. Okay, so base pairing. Or in other words, we can write 10 to the minus 2. Okay, so there's a second sort of related pair of properties that actually have to do with the active site of the DNA polymerase. So it's related to the active site of the polymerase. And this is referred to as shape discrimination. First, shape discrimination refers to the fact that in the polymerase active site, incorrect bases will not fit precisely, or fit much less precisely than the correct base. And the second sort of part of it is that if by chance an incorrect base finds itself in the active site, it's not in the appropriate configuration for optimal catalysis. So there's sort of two components to this active site of the polymerase. The wrong base doesn't get into the active site very well, and even if it does, the reaction with the wrong base proceeds less efficiently than the reaction with the correct base. And this contributes another sort of 1,000-fold. Okay, so in the active site, just to reiterate, incorrect bases don't fit precisely. And if it does, it's not optimal. For base incorporation. And again, as I said, that contributes another 1,000-fold, or 10 to the minus 3. Okay, so that would take our error rate, if we add 10 to the minus 2 plus 10 to the minus 3 to now standing at 10 to the minus 5. And interestingly enough, this is actually the same error rate of RNA polymerase. And we'll see that later on when we delve into the RNA section. Okay. All right, so we have those two components, the base pairing and the shape discrimination, at this point now contributing to 10 to the minus 5 in terms of fidelity of the DNA polymerase. And then if you recall, we mentioned in the previous slide that the DNA polymerase has this mysterious 3' to 5' exonuclease activity. And what it actually does is it's capable of removing incorrect bases. So if at the shape discrimination stage you still get an incorrect base that's actually polymerized, there is one more opportunity to get rid of it at the proofreading stage. So really this is due to the 3' to the 5' exonuclease activity. So we can remove the incorrect bases if they're still incorporated. Okay. So this raises things up about another 100-fold. So about 10 to the -2. And now we're up to, if we add all this together, 10 to the -7. And this is really sort of the intrinsic fidelity of the DNA polymerase itself. So the intrinsic fidelity of DNA polymerase. And then in cells, there's also contribution of a DNA repair pathway that makes up an extra 1,000-fold difference. So there's this repair pathway called mismatch repair, as I've outlined here, which removes base mismatches subsequent to DNA replication. So it removes base mismatches subsequent to DNA polymerization. And as I mentioned, it's 1,000-fold, or 10 to the -3. Okay, so that accounts for, if we add the 10 to the -7 plus the 10 to the -3, this actually gets us to 10 to the -10. So now let's have a look at the enzyme itself. So once the enzyme was purified and probably more importantly produced in recombinant form and purified in larger quantities, it enabled the determination of the structure of the DNA polymerase. And its structure, like many things in biology, is described as a hand. So it's described as a hand. I guess, you know, structural biologists they like things to look like hands, but at any rate, the idea is that it's a sort of a multi-lobed, multi-domain structure that we see in this top figure here. So in this one right here, it sort of has this platform, which is shown in blue, and which is often referred to as the palm. Okay, so as the palm, as it's stated here. And then it has these gnarly fingers, which are shown in yellow. So up here. And the active site for polymerization is kind of illuminated in white here. Okay, so active site. So AS is the active site. And then there's this sort of junction between the fingers and the palm. And the fingers are important for recognition and binding of the incoming DNTP. And this thumb that's hanging off here in green is important to forming this channel that the DNA template resides in. So there's a channel. So forms a channel for the DNA. And then at the bottom here is the exonuclease domain. So that's in red. And again, the active site we've shown in white. So what's interesting about this is that the enzyme actually has two active sites, right? One for polymerization, which is up here in white, and then we have the exo site, which is down here, and again, that's for proofreading. So the polymerization site and the exonuclease site are distinct, and I guess one consequence or thing that sort of agrees with this type of setup is that the reactions are also distinct. Down here, this is actually the exonuclease reaction. So the exonuclease reaction, and it is not the reverse of the polymerization reaction. Which is this one here that we saw in the previous slide. So it's obviously not the reverse, right? If it were, we would consume a pyrophosphate, which it does not, and it would release a nucleoside triphosphate, which it also does not. So two distinct active sites, two distinct chemical reactions catalyzed by them. So you might ask, how does all of this work? If we have an active site that polymerizes and then an active site that degrades, so how do you ever actually get anything done? So you're like running like a hamster on a wheel, polymerizing a nucleotide, exonucleolytic cleavage of a nucleotide, and so on and so on and so on. Well, the way it works is that the reaction rates are very, very different at the two sites. Okay. So the reaction rates are different. And as you might imagine, the reaction rate of the polymerization active site is much, much greater than the exonuclease site. So the polymerase activity is greater than the exonuclease activity. Okay, so polymerization is greatly preferred. So the 3' end of the DNA primer spends most of its time in the polymerization site and not in the exonuclease site. The one exception to this is if you have an incorrect base incorporated into the DNA, that can cause a distortion in this channel that the DNA is leaving through, which then increases the likelihood that the 3' end of this thing, the newly synthesized DNA, will find itself in the exonuclease site. Okay, so if we have something that's incorporated, if we look at this figure in the active site, if it's an incorrect fit, it will cause a distortion in the polymerase and then move this 3' end into the exonuclease site. Okay, so just to reiterate, the 3' hydroxyl of the primer spends most of the time in the polymerization site. If a mismatch occurs, it causes a distortion in the DNA polymerase, and it moves to the exo site. Okay. And when it does find itself in this exo site, then it's subject to removal of a nucleotide. Removal of this mismatch then sort of restores the normal shape of this channel, and then again prefers polymerization. So you can kind of, I don't know, this is maybe a bit hard to see, but in this crystal structure here at the top, there's actually DNAs from two different structures overlaid on each other. One of them has the 3' end, this red one, in the polymerization site, so up here. So this is the poly site. And then this sort of blue-green thing has the 3' end and the exonuclease site. So this is the exo site. So in this configuration, at the top you would be polymerizing, and in this configuration down here below you would be proofreading. Curiosity about the journey to the purification of DNA polymerase 1. As I pointed out, we saw that the enzyme had a few shortcomings that didn't seem to be consistent with it being capable of replicating an E. coli chromosome, and that the enzyme didn't polymerase particularly rapidly, nor did it polymerize processitively. And so, although Kornberg and his colleagues were definitely working under the assumption that this was the DNA polymerase that was doing the DNA replication of the genome in E. coli cells, because it was the only polymerase that they detected. So they were working under the assumption, or they assumed, it was the one that polymerized. The genome, since it was the only one detected. Okay. John Carnes, however, as I've named him here, however, he was not convinced by their argument. So he really set out to determine that DNA poly 1 was not the replicating DNA polymerase. Okay. So the first, really, the counter argument, of course, was this processivity thing, that it simply just wasn't fast enough to account for how many nucleotides E. coli polymerase had to synthesize during one cell cycle division of the bacteria. And then secondly, the estimate of how many molecules per cell of DNA polymerase 1 existed was around, or determined to be around 400. Yet the E. coli chromosome is replicated by 2 replication forks. So if you only need, for argument's sake, 2 polymerases per replication fork, that would be 4. So how come there's like 100-fold excess? So that didn't make sense to Carnes either. So he sort of kind of had this twisted experiment to show that DNA polymerase 1 that Kornberg had identified was really not the replication polymerase in E. coli. And his reasoning, it was along those lines. So, we know DNA replication is essential for life, right? A cell can't divide if it doesn't replicate its DNA. The DNA polymerase that replicates the DNA, therefore, must be essential for life. So the idea is, if you had a cell that lacked the polymerase, then it would not be able to replicate. Its DNA would not be able to grow and divide. And so he sort of turned that on its head and said, let's look for a mutant E. coli that does not have this DNA polymerase 1 activity that Kornberg identified, yet is still alive. So you have to have a certain amount of confidence in your hypothesis if you're going to go down this road, right? Because if he was wrong, they would never find it. So I don't know. It's kind of an interesting approach. So this search was actually done by his laboratory assistant, Paula De Lucia. Okay. And what she did, she sort of started with a kind of library of literally thousands of mutagenized E. coli strains. Okay, so she started with a library of thousands of mutant E. coli strains. Okay. And then she assayed them for DNA polymerase activity. All right, so Paula literally started with this kind of library where she had thousands of these mutant E. coli strains. She took each of these strains and then grew them up, made an extract according to the Kornberg protocol, and then assayed them for DNA polymerase activity according to the Kornberg protocol. And she was looking for one that did not make DNA, so this is like serious torture, right? So she did this for a long time, and then on the 3,478th extract, so the 3,478th extract, she actually found one. Okay, she found one with no DNA polymerase activity. Okay, thereby supporting Karn's hypothesis that DNA polymerase 1 is not required for life. So therefore, DNA poly 1 is not required for life. Okay. Therefore, it can't be the polymerizing enzyme that's replicating the chromosome. So the gene for DNA polymerase 1 gets called poly A. So DNA polymerase 1 gene gets called poly A. And again, get it for like poly A. So that's kind of neat. So as a result of this, Kornberg kind of goes into this deep depression. The enzyme that he had staked his fame on was determined not to be the enzyme that replicates the E. coli chromosome. So he's very, very, depressed by this discovery. However, the existence of an E. coli strain that is a poly A mutant now gives you a tool to find other DNA polymerases that might not be as abundant. So you can basically make an extract from your poly A mutant and just kind of turn the wheel, assay that for extract for polymerase activity. You find a very small amount, and then you proceed with the same fractionation kind of scheme that we discussed before. And if you do that, the first thing you come up with is an enzyme that was named poly 2. So they found another polymerase. It was called poly 2. Sadly, this is also not required for life. Okay, so not required for life. So it wasn't until poly 3 was identified. So then they found poly 3. That the actual enzyme that replicates the E. coli chromosome was found. Okay, so this is the replicative enzyme. And just sort of to complete the circle, the person that discovered and purified poly-3 was a graduate student named Tom, but his last name is Kornberg, so he's actually Arthur's son. So I guess it's like a family thing, I guess. Okay, so this was discovered by son of Arthur. Okay, so there's a couple of things you can take away from this, other than, Paula had incredible patience and fortitude, is that even a pathetically simple cell like E. coli has more than one DNA polymerase. So polymerases play multiple roles intercellularly and have become specialized such that different polymerases can fulfill different tasks. So there are specialized polymerases that participate in DNA repair rather than replication. There are in eukaryotic cells polymerases that function in specific compartments of the cell, as we'll see later on. So for example, there are some polymerases that function in the nucleus. There is a DNA polymerase that functions in the mitochondria to replicate the mitochondrial genome. And even at the replication fork in eukaryotic cell, the role of DNA polymerases has become specialized such that although E. coli chromosome is replicated by polymerase 3, if you actually go to a eukaryotic cell, we actually require 3 DNA polymerases, and they include DNA polymerase alpha, DNA polymerase epsilon, and DNA polymerase delta, which we'll see later on. So DNA polymerases fulfilled more than one role. They have some degree of specialization, but despite this, they all kind of look the same. Okay, so you have this sort of palm platform, these gnarly fingers and the thumb, and ones that actually proofread. Not all of them, however, do have an exonuclease domain. So poly-1, despite not being the replicative polymerase, is indeed sort of the conceptual prototype for all DNA polymerases. Okay, so just write that down. Poly-1 is really the prototype for all DNA polymerases. And on this slide, we have all of our stars. Carnes at the top right was a man that was not prepared to accept Kornberg's assertion regarding DNA polymerase 1. Paula on the top left, she did all the work with all those thousands of mutants of strains. And then below, Tom Kornberg, who eventually purified poly-3. All right, so what I kind of like about the whole DNA polymerase thing, and I'm gonna preach here for a moment. So basically, there was a bunch of scientists in a bunch of different labs, and their goal was to figure out how the DNA polymerase worked, right? This was this super interesting DNA structure that was proposed. It was really clear that by the time that DNA was the genetic material, when they discovered that, they wanted to know how the thing was copied. Basically, the goal, they were just trying to figure stuff out. So if you look, this was in 1956, so you look downstream, years downstream of this, lots of stuff does get enabled by it, right? I mean, you can never sell anyone on the idea of funding people who are just trying to figure out stuff, from a financial perspective. Yet downstream, it's pretty amazing the material that actually comes out of it. So 2 technologies that depend on the understanding of how DNA polymerase how it works is, super easy. So, one of them, of course, is PCR. So how much is PCR worth? So PCR was originally patented by CETUS. So CETUS patent PCR. And then they sold it to Roche. And that was sold for a cool $300 million. So a tremendous amount of money. Then the patent went on to actually earn $12 billion in royalties. Before it went off patent. So that's pretty good, right? Another super obvious technology. So what do we do if we want to know the sequence of our DNA? We apply DNA sequencing, right? So almost all sequencing methodologies are sequencing by synthesis. So, they are barely glorified DNA polymerase assays, right? So we have the sequencing market. So number one was PCR, so sequencing. So again, you can think about it, if you have no DNA polymerase, you have no sequencing. So the sequencing market, I can give you some numbers. In 2013, was worth 1.1 billion. In 2017, that went to 4.1 billion. And estimated for 2025, it'll be worth a cool 22.7 billion. So, huge amounts of cash, 30 years or 40 or 50, 60 years after the fact, that flow out of scientists just being curious about how things work, right? And then, genome editing is another super good example of that right now too, right? So we can include genome editing. So there's a lot of foundational genome editing work that, that come from scientists just trying to be able to figure stuff out. So really, the key thing I want you to take away from this slide is really don't underestimate the power of just how understanding how things work and learning about how they work, how important those two things are in research. All right, so what we're going to do here is actually just take a step back and try to really incorporate all the information that I've presented to you. And just imagine yourself being asked to set up a DNA polymerase assay. And the two things I want you to think about are, what would you include in this assay? And the second thing, how would you determine if any DNA has been synthesized in this assay that you set up? Okay, so we want to set up a DNA polymerase assay. What are we going to put in it? So what goes into this polymerase assay? Well, obviously we are going to need DNA polymerase, right? That's a given. So we need the enzyme itself. What else do we need? We also need the DNTPs as our substrates, right? So the DNTPs, and we'll need all four of them, all right? What else do we need? Do we need ATP? So that's something to think about. Kornberg needed ATP, right? But we don't actually need it because the DNA polymerase itself is actually using energy from the hydrolysis of the DNTPs, or cleavage of those phosphate bonds, in the Kornberg setting, the ATP was actually kind of important for the kinases that were phosphorylating the nucleoside to monophosphate, diphosphate, and triphosphate. So in a DNA polymerase assay per se, we actually wouldn't need to add ATP. We will, however, throw in the magnesium for fun. Okay, So we'll put in magnesium because DNA polymerase loves magnesium. So what else is needed? We've got to copy something, right? So we're going to need a template. And what do we need annealed to our template? So we need a primer, as we discussed before. So really those are the bare bones components of a DNA polymerase assay. So these are all our substrates. That's really all you need. And then it's also good if you can tell that you've made DNA though, right? So how do we tell if we've made DNA? You could sort of obviously use the radiolabeling approach, which is always good. That's what Kornberg used. So remember, he radiolabeled the DNTP. So there's, like, in theory, radiolabel the DNTPs. So attach a radiolabel here. So radiolabel. Also in theory, you could radiolabel the primer. Okay, so you could have a radiolabeled primer. And really the radioactivity is a good means of following incorporation of nucleotides into a polymer, and then of course you need some kind of separation methodology, right? That will distinguish your nucleotide that's unincorporated from your polymer that you've synthesized. So in the case of Kornberg, he used acid solubility. In a more modern setting, we might want to use gel electrophoresis. So for separation of nucleotides from polymer, we can use gel separation. So for example, to separate very small things like nucleotides from very large things like the synthesized DNA. So we have the enzyme, the substrates, and then of course you need some sort of means of detection. Okay, so we've set up our DNA assay. All right, so kind of moving right along here. So that's the kind of the in vitro DNA polymerase material that we've just gone through. What I want to do now is to actually turn to a couple of experiments because we know we are sort of historically, we've got this model in which DNA is unwinding to provide a template for its replication. And now we've identified an enzyme that can synthesize DNA using an existing DNA strand and primer. But the question now becomes, how does this actually, what is the process that is actually carried out in a cell? What are the properties of replication that we can't really account for with the in vitro information that I just presented to you? And what might that be, and how we're able to examine it in a cellular setting. So what's to follow is 3 sets of experiments, and I will definitely we'll start off with the one that demonstrated semi-conservative replication, and we'll delve into bidirectional and semi-discontinuous in the upcoming lecture. The experiment that actually demonstrated that replication is semi-conservative in cells was performed by Messelson and Stahl, and this, again, as you can tell by the date, was in the late 1950s. So there's a lot of stuff in the molecular biology world converging around DNA replication during this time. And I think it's one of the most beautiful and sweetest experiments in all of molecular biology. It's first very super well organized. It second asks a very direct question. And proposes, thirdly, as you know, they performed a very meaningful experiment, really directed at clearly distinguishing between the possibilities that they imagined. So in thinking about DNA replication, they were thinking about three possibilities. The first was this semi-conservative model that you start with, your parental DNA strands. The two strands dissociate from each other. Each one is a template for new synthesis, and those remain as the products of the reaction. So the products of the reaction have a parental strand and a new strand, and again a new strand and a parental strand. So you have your parental plus your new strand. Okay, and this is the semi-conservative model that was actually proposed by Watson and Crick. So this was proposed by Watson and Crick. So that was the first possibility. The second was the semi-conservative reaction, that perhaps these strands come apart and serve as templates, but the original parent strands actually come back together, and the daughter strands that are synthesized, they come back together as well. So this would be a conservative mode, and this was proposed by Max Delbruck and Gunter Stant. Okay, and then a third model again was proposed by Delbruck, and it is this really messy random dispersive model. And Delbruck's thinking was that the energy holding the double helix together was potent enough that he felt like it was highly unlikely that you could separate those strands without causing breaks in the backbone. So in the act of replication, he imagined that you would have to break these strands into pieces, and then you'd end up with these sort of tracks of old and new DNA sort of mixed up. So these are the three things that Messelson and Stahl were proposing to distinguish between the three possibilities for DNA replication. So how did they do this? All right, so the first thing they did is, how? They started with E. coli. And they grew the E. coli in the presence of a nitrogen source that was N15 rather than the conventional N14. And the great thing is that the DNA results in having a higher density because N15 is more dense than N14. So N14. Okay. So really, this really gives them a means of distinguishing, old DNA from, say, new DNA that would have the N15. So they would actually grow these cells for many generations. In N15. And then transfer back to N14. And they would sample at different times. Okay, and after they sample, they centrifuge this DNA. Okay, so they would centrifuge. And they would centrifuge based on density. In a density gradient. And really the density gradient separates on the basis of density. So it kind of looks like this, where you have a tube, and then there's some liquid, which is a solution of cesium chloride. So cesium chloride. And the cesium chloride is more dense at the bottom, than at the top. Okay. And then if you layer your sample at the top, so you put your DNA here, and then you centrifuge this. The DNA will actually migrate into the density gradient, and it will stop at the position where the DNA density is equal to the gradient density. So you would get a tube that kind of would have, different things within it, and it would stop, your DNA would stop where the DNA density is equal to the cesium chloride density. And if you have DNA species of different densities on this gradient, they'll migrate at different positions. So, here's your DNA fragments. All right, so that was their setup for determining how replication is carried out in the cell. Okay, so I'll introduce you to the slide, which will continue on in the upcoming lecture. So the only thing I want to say for this figure right now is that basically this is a side view over here of the density gradient. And if we focus in on here, it is at generation 0. So this would be the completely N15 species of DNA that we will be looking at. And as we can see, it actually just gives us one species, which you can detect because DNA absorbs UV light. So this is the one band in their input DNA, and when we start in the upcoming lecture, we'll discuss what happens as they sample during the growth and division of the cells over the course